The future of targeted therapies lies in precision. Antibody-drug conjugates embody this therapeutic precision with their intricate structures, which unlock high potency. But this complexity requires specialized process development, analytical insights, and manufacturing experience. At Samsung Biologics, our approach goes beyond simply transitioning from one step to the next. We advance molecules into medicines and turn insights into measurable impact, transforming potential into real therapeutic possibilities.
ADCs vary in payloads, linker chemistries, and conjugation method, so a generic approach will not meet diverse project needs. Thus, for the process development, we create bespoke, data-driven strategies using systematic parameter screening, such as design of experiments, to optimize key conjugation variables like reduction conditions and quenching processes. For the drug product, we conduct various analytical testings and stability studies to finally select the formulation and the lyophilization cycle that are specific to the client's ADC. Our capability spans flexible services. In terms of drug substance process development, we provide from small 2 milliliter feasibility studies to commercial scale manufacturing using 500-liter single-use or stainless steel systems. Early definition of critical parameters based on reproducible data ensures seamless integration across development, tech transfer, and manufacturing stages. We also support a broad range of conjugation types, random cysteine, lysine, and site-specific methods, covering a wide spectrum of payloads like DM1, DM4, MMAE, and DXD. Similarly, our drug product development services provide not only the SBL DP platform services, but also DP services with customized study design that meet the needs of our clients. Regardless of the dosage form selected by the client, whether the frozen liquid or the lyophilized solid, we maximize our expertise and communicate closely with our clients to provide tailored DP services. We also conduct antibody and ADC development in parallel, compressing project timelines from conceptualization to GMP manufacturing.
The heterogeneity of ADCs demands thorough, multifaceted analysis. Our analytical team combines bioconjugation expertise with a comprehensive suite of techniques including LC-MS, HIC-HPLC, RP-HPLC, SEC models, UV-Vis, CSDS, gas chromatography, as well as cell-based assay. These methods enable accurate measurements of drug-to-antibody ratio, conjugation sites, free drug content, residual solvents, purity, as well as potency. Case studies highlight dark consistency within ±10% of the target and 96% or more monomer purity across various scales and multiple linker payloads. We develop rapid, project-specific analytical methods early on to support process understanding from the start and data-driven decision-making to accelerate development timelines without compromising accuracy.
ADC tech transfer involves unique chemistries and potent compounds requiring highly specialized controls. We design client-specific, validated transfer strategies prioritizing critical quality attributes such as the drug-to-antibody ratio and residual solvents verified by orthogonal methods like high-resolution LC-MS. Our seamless tech transfer approach spans early clinical through IND phases, shortening timelines while preserving quality and compliance. Leveraging extensive monoclonal antibody and mRNA manufacturing experience, we standardize material handling, including specialized conjugation bags for highly potent API management to ensure stability and precision. Samsung Biologics emphasizes strong client-centered communication that links strategic design to reliable large-scale and phase-appropriate production. This ensures transparency, alignment, and proactive issue resolution from early development through commercial supply.
We operate a purpose-built end-to-end ADC manufacturing facility designed for highly potent API handling. It is co-located with our monoclonal antibody sites to maximize supply chain efficiency and minimize risks. Our flexible conjugation trains support both random and site-specific conjugation technologies, and we can offer scales up to 500 liters. To ensure safety and product quality, we have implemented a stringent containment and training strategy. This has been confirmed by surrogate testing so that we can operate as low as an OEL of 5 nanogram per cubic meter. Our proven infrastructure and process deliver consistent quality while adapting the evolving client needs, from conjugation through purification to final bulk drug substance manufacturing. Our investment in ADC-DP capabilities will offer the next step of your end-to-end service. We provide a fully integrated ADC development and manufacturing service, linking your innovative ideas to manufacturing reality, your science to patient benefit, and linking your antibody to its fullest potential.